Greetings everyone. We are from Group 9 and our project is regarding skin cancer AI. Myself Tanmay Garg and I have majorly contributed towards setup and configuration of hardware part of our project.
I am Aditya Chawla. I developed the computer vision model for classification.
I'm Akshit Gupta. I have primarily worked on research and data gathering.
My name is Avneet, and I have worked on hardware implementation and setup of the project.
My name is Akarshan, and I have worked upon research and data gathering in this project. Now, I would like to hand it over to my teammate to explain the workflow of this model.
Okay, so here we are where I will explain how our project works from the very start to the very end. Our primary goal for the project was to detect skin cancer tumor in earlier stages so that we could increase survival rate of a particular individual. In order to achieve that, we basically divided the workforce of our team into three major categories. The very first one being research and dataset gathering, second is hardware implementation, and third is software implementation. Moving on further, here is the flow chart of all the different timelines that our team members have been working on synchronously in order to achieve at the very last our main goal. The very first timeline consists of dataset gathering that we have used from a website called Kagan. And after that, we process the data and perform cleaning operations in order to generate a training model and a validation accuracy that is satisfying to us. The test accuracy is tested, and if the accuracy is satisfied to us, then we'll deploy the CNN model. Else, if it is not, then we'll again train the model again and again until and unless we achieve a particular minimum threshold of the satisfying accuracy. After this accuracy has been achieved, we are deploying our CNN model. Here deploying by, I mean, we are converting our Python file to PB file. Here PB is nothing but a frozen graph file. In frozen graph, all the weights and biases of a machine learning model are frozen and you can't alter them. Now, moving on to second timeline, configuration of virtual machine is done using Ubuntu 18 as our operating system. After that, we are configuring OpenVINO toolkit that is required in order to communicate with Raspberry Pi and neural compute stick with the help of machine learning model. After that, we are using model optimizer alongside the CNN model that we have built before in order to generate IR representation of the model. Now, IR representation will give us three files, XML, mapping, and bin. Out of these three files, XML and bin are used alongside with the neural compute stick to generate the output. Moving on to third timeline, configuration of Raspberry Pi 3B Plus is done. We have installed Raspbian Buster operating system over here. Again, configuration of Open Toolkit is done here. Side note, configuration of Open Toolkit in this particular timeline is completely different in way from compared to our configuration on virtual machine on the PC. After that, we are adding NCS2 USB rules. NCS2 USB rules alongside with other setup and optimizations are required in order to work neural compute stick with the required hardware and software synchronously. At the very last, we are detecting skin cancer alongside using an API which is classification sample.py with an image that we will be detecting if it is either benign or malignant alongside with our neural compute stick that will do the heavy lifting. All these three are combined alongside with our model, and in a command prompt we will get the answer with the confidence printed among beside them, that is benign or malignant. So that was the brief explanation. Now I will hand over to other team members so that they could show you the live working of our project.
We will be using Google Colab as a development platform. We start by mounting the drive, then we import the relevant libraries. Next, we load the dataset, which consists of 2000 plus images of skin lesions. Then we select the module for transfer learning using MobileNet V2. Then we set up our data generators, which augment the images before feeding them to the neural network. The sequential model consists of a linear classifier on top of a MobileNet V2 feature extractor from the TensorFlow Hub module. Then we train and validate the model to achieve an accuracy of 82% after 10 epochs. We plot the accuracy and loss curves to analyze the performance of our model. Then we move on to understand the effectiveness of our model by analyzing certain metrics like F1 score, precision, and recall from the classification report. We can further visualize these in the confusion matrix. Now we test the model on previously unseen images. Finally, we save the model as a frozen graph and download the .pbtxt file. That was a brief explanation of how the software was developed and plays role in our project. For hardware component, I will let my teammate take over.
So I'm going to give a brief explanation of our hardware implementation. This is the hardware used in our project. Right here we have Raspberry Pi, NCS2, camera, and SD card. SD card acts as storage. The Raspberry Pi is the SPC used in our project. NCS2 is the VPU, which is main brain of the project. Camera is used for collecting data. So after being connected to a monitor, we will create this folder which will contain all the images the frozen graph and the IR representation. So this API right here is used, acts as bridge between IR representation and images. So first I will take an example by running image which is malignant. As we can see, we have reached 95% probability that the image is malignant. Now I will take an example of a benign image. We have reached 99% probability that the image selected is benign. So this proves that the VPU used is working exactly as intended and classifying images. Thank you.